Hello and welcome to Physiology. This is Chapter 1, and in Chapter 1 of our class, we take a moment to talk about what physiology is and to review the various components of the body. We begin with Section 1.1, the scope of physiology. Physiology is the study of how living organisms function. Our class is human physiology, so we focus on human systems, but much of the knowledge that we've gained regarding physiology over the years has come from living organisms other than humans, and periodically during the course of our class, we'll make reference to those studies. The scope of physiology is extremely broad, and we'll touch on many of these different aspects of physiology during the course of our class. For example, on one end of the spectrum, we have essentially molecular physiology, and so it really all begins with the interaction of those molecules with ions, for example, in the nervous system, and the interaction of molecules with each other when we talk about the endocrine system. And then at the other end of the spectrum, we have functioning of entire systems. For example, the cardiovascular system, circulating blood through the body, or the respiratory system, moving air and then oxygen into the body and then into the blood and eventually to the cells. And so our physiology course will touch on different aspects of physiology from very small down to molecular level all the way up to the entire body system. Your book points out something that's very important to remember, that physiology is studied at many different levels, and most of these studies are actually at some level below the actual organism. And so most physiologists are working, for example, in cell culture or in molecular studies. Some are working in mouse models as well. But your book points out that when we do these physiological studies, our goal is to take the information that we gain and then apply it to the entire organism. But bit by bit, through these smaller studies, we gain information regarding the physiology of the entire organism. Some areas of physiology are still poorly understood. Some areas are quite well understood, the cardiovascular system perhaps being one. We'll spend a lot of time in the cardiovascular system during the course of our semester. But other areas, especially in the nervous system, particularly the brain, are relatively poorly understood. We have 86 billion neurons that compose our brain, and that's just a gigantic number of cells, and we have an incomplete understanding of how they interact with each other at this particular point in time. If we look at the last point on our slide, pathophysiology, what we see is this, that pathophysiology is the study of disease and when normal homeostasis is disrupted, and then what we can learn from that disruption. So it's a bit morbid, perhaps, but it is true that through the study of pathophysiology, we can gain a great deal of information about physiology itself. One example is the famous painting that we see on the screen. This is a poor person suffering from tetanus, as you likely realize. And so tetanus is a disease that we'll talk about the molecular mechanism of. It's a disease in which the snare proteins at the neuromuscular junction are damaged, preventing the release of inhibitory neurotransmitters that cause the relaxation of muscles that serve as an antagonist. So we'll talk about that in more detail later in our class. But for now, we want to simply make note of the fact that pathophysiology is the study of physiology gone wrong. And we do, at many points during the course of our class, make various observations and notes on pathophysiology. To be honest, when we think about pathophysiology and many of the examples that we'll talk about during the course of our class, many of those examples are toxins that are produced by one organism and inhibit some functioning of physiology in a second organism. Very often, as I mentioned, the nervous system. So pathophysiology, historically, and even to this day, has been very important in helping us understand physiology as a whole. We move on to section 1.2, and in section 1.2, we start to talk about the organization of the body, beginning with the smallest units of living units of organization in our body, which are cells. So our body consists of a giant number of cells, 86 billion cells in the brain alone. And if we look at the different types of cells we have in our body, we can categorize perhaps 200 based on their shape and their function. However, all those cells can be put in larger categories that essentially boil down to the four categories that we see on the screen. these categories correspond with the four tissue types that we'll talk about momentarily. So the four broad cell types that we possess are muscle, nerve, epithelial, and connective. Now, cells are living units. Cells can be removed from the body and survive for a certain period of time. Conducting research in cell culture is a very common way of conducting research. This is not a type of research that I do anymore, but some time ago, back in grad school, part of my research was cell culture, and so we would do primary cell culture. There's essentially two types of cell culture. One is primary, in which, like we did, we took livers and dissolved those livers, and then you could grow the hepatocytes in culture for about 2 weeks and perform experiments on those cells. The other type of cell culture involves utilizing what are referred to as cell lines. And so these are typically cancerous cells that have been modified. These are referred to as immortal cells because they divide and divide and divide and live forever, not one cell, but of course, as it divides, it produces offspring that survive. Of course, the HeLa cells are a very famous example of this. But in any case, cells are the smallest living units of the body, and we have four cell types. Some of our discussions during the course of our class will focus on cell physiology. We move on to tissues. And so a tissue is defined as a group of cells that work together to perform a common function. In the body, in the human body or any vertebrate body, we have a maximum of four tissue types: muscle, nerve, epithelial, and connective tissue. So what we see is that those tissues are groups of cells. Now there are not uncommonly different types of muscle cells, different types of epithelial cells that are joined together to make a tissue, but nonetheless, there's just one cell type overall that's found within that tissue. We move on then to organs. And what we see is this, that organs are composed of multiple tissue types, and typically an organ contains all four tissue types. And so if we think of a simple example, we can think about the heart. The heart, by weight, consists mostly of muscle tissue. But if we look closely at the heart, and as we'll talk about in detail during the course of our class, there is, of course, something referred to as the conducting system of the heart that begins with the SA node in the right atrium. This is nervous tissue that's part of the heart. This imparts the autorhythmic activity of the heart, allowing the heart to beat. As you are aware, your heart beats independently of your brain. The brain can adjust the speed of the beating of the heart, but it doesn't ultimately initiate the heartbeats that allow, or the electrical signals that allow the heart to contract. If we go a little further, what we see is that we have epithelial tissue that's found both on the outside of the heart and on the inside of the heart. We'll talk about the pericardium and the epicardium when we get to the cardiac system, cardiovascular physiology chapter. And lastly, we have connective tissue that makes up a portion of the heart. And so, for example, the fibrous skeleton of the heart is composed of connective tissue that joins the atria on roughly the top of the heart to the ventricles found on the lower portion of the heart. Some of our time during the course of our class will be spent talking about organ systems. And so, in fact, our class, to some extent, is organized by these organ systems. For example, what I just mentioned, the cardiovascular system is one chapter of our text. Our respiratory system is one chapter of our text. The endocrine system is another example of an organ system that we'll talk about during the course of our class. And so organ systems consist of a number of organs that work together to perform a common function. Now, in some cases, that's pretty simple, like in the case of the cardiovascular system. We have a heart, we have blood vessels, and everything is fairly straightforward. In other cases, it's a little more complicated. And so what we notice is this, the endocrine system, some students sometimes express frustration with the endocrine system because it's not a discrete system. And what we see is this, we have some glands that are part-time endocrine glands, the pancreas being an excellent example. And so the pancreas is an exocrine gland, a definition that we'll cover soon. Exocrine meaning it secretes something into a duct or a tube. It's most of the tissue by weight is exocrine, but it also serves as an endocrine gland in the body, of course, producing insulin. When we look at muscle cells, we see that muscle cells are only capable of doing one thing, and that is decreasing in length or decreasing in overall size. So, interestingly, each of the three types of muscle cells do that in slightly different ways that we'll talk about during the course of our semester. And each one possesses some differences in terms of their overall anatomy, and those differences in anatomy are linked to their differences in physiology. If we begin briefly with skeletal muscle, we're of course aware that skeletal muscle is the one type of muscle that is under voluntary control. Skeletal muscle is always found attaching either bones to bones or attaching bones to skin. And so, of course, when that skeletal muscle decreases in size, it causes movement of one or perhaps both, in some cases, of the attached structures. We have a nice diagram on this particular slide that shows us the overall organization of skeletal muscles. And as we go through the course of our semester, we'll talk about this in greater detail. But we do see that skeletal muscle cells, very briefly, are made up of myofibrils. If we start at the right-hand side of our diagram, we can see a single myofibril. Those myofibrils go together to make a single muscle cell. We do notice that muscle cells are sometimes referred to as muscle fibers as a result of the fact that they are extremely long cells. You'll see in that same diagram that there's a bundle of muscle cells that are together there in something referred to as a fascicle. Our diagram is labeling those as muscle cell bundles, also referred to as fascicles. There's connective tissue that surrounds those fascicles, and then there's an overall outer sheath of connective tissue that circles all the encircles all the fascicles. If we go now to cardiac muscle cells ever so briefly, we really don't talk about their anatomy at this point, but we do perhaps would perhaps like to make a note at this time that cardiac muscle cells are much shorter in length. Cardiac muscle cells are still striated. We actually didn't mention that skeletal muscle cells are striated. They have alternating dark and light bands. We see the same thing in cardiac muscle cells. And we see that cardiac muscle cells and skeletal muscle cells have different proportions of the proteins that we haven't discussed yet and won't in this chapter that allow muscles to contract. If we go to the last type of muscle cell, we see that we have smooth muscles. Smooth muscle cells, are common in many of the tubules of the body. Blood vessels and our airways are examples of such tubes. And smooth muscle cells are not under voluntary control, as we well know, and they're also not striated. They do possess myosin and actin in an arrangement that we'll talk about in that particular chapter, the muscle chapter, but they do not have the same striations and Z-disc and so forth that we observe in the skeletal muscle and cardiac muscle. So, interestingly, this will be our overview of muscle cells for the moment until we get to that chapter later in our class. We spend just a moment talking about neurons at this point in our class. Neurons actually only do one thing as well, and that is they are able to conduct electrical signals and in some cases initiate those electrical signals, in some cases over long distances. Typically, a neuron is connected to one of three potential so-called postsynaptic cells. A synapse is the junction between a neuron and whatever comes after that neuron. And so we see that we typically have three postsynaptic cells. One possibility, and this is by far the most common, is that the neuron stimulates another neuron, and so it connects to a second neuron. We see a little picture on our screen of neurons connected together. We do see that the majority of neurons that are found in the brain are referred to as interneurons. These are neurons that are connected to one another and not to some other one of the other two effectors that we'll talk about next. Another possibility, though, and this is not as common, is that a neuron stimulates a gland cell. And so we see this in the case of the adrenal medulla as it releases adrenaline. And we see it rarely in other cases in the body. In the pituitary, posterior pituitary, we see it as well, as we'll talk about. But that is one possibility. A second possibility, that a neuron stimulates a gland cell. And then a third very common possibility is that a neuron stimulates a muscle cell and causes that muscle cell to contract. And so we'll spend a good deal of time talking about that during the course of our class. But we do see that without exception in the body, skeletal muscles contract as a result of the termination of a nerve cell upon them. We also see that smooth muscle contraction is regulated. Not the contraction is not initiated, but the speed of the contraction and strength of the contraction in some cases regulated by nerves that terminate on those smooth muscle cells. What we see is the following, that we use the term nerve to describe neurons that are packaged together. Specifically, most often nerves are these so-called axons, extensions of a neuron that may travel for a long distance through the body that are packaged together. We have a large number of neurons that are found in our nervous system overall, 86 billion, as we mentioned earlier, just found within the brain. We now look briefly at epithelial cells. And as we previously discussed, epithelial tissue is a type of covering and lining tissue. We find it covering the outside of the body in the form of the skin, and we find it in various locations inside the body. We already talked briefly about the epithelial tissue that lines the inside, for example, of the heart. So epithelial tissue is specialized for protection in some cases. Our skin would be a great example of that, protecting us from bacteria, from desiccation, in other words, drying out. But in other cases in our body, our epithelial tissue is specialized for either selective secretion or selective absorption. So we certainly see that in the case of our kidneys and our digestive system. What we notice is the following, that epithelial tissue is characterized based on the shape of the cells and the arrangement of the cells in space. And so as we look at this diagram from our text on the right-hand side of our slide, what we notice is the following, that we have three different cell shapes that may be found in epithelial tissue, cuboidal, columnar, and squamous, and those cells may be arranged into these different arrangements that we notice over on the right-hand side of the slide. Principally, they may be simple, in which case we have just a single layer of cells, or they may be stratified, in which case we have multiple layers of cells. So a couple quick examples. We see, starting at the top on the right-hand side, simple squamous epithelium. Simple squamous epithelium is found, most notably perhaps, lining the inside of our circulatory system. This includes the inside of the heart, the inside of the blood vessels. In fact, it makes a continuous protective layer, and our blood never comes in direct contact with our vessels. It's always separated by this simple squamous epithelium. If we make our way down one box, we see simple cuboidal epithelium. Simple cuboidal epithelium is found lining the tubules of the kidneys, where it's specialized for secretion and absorption. If we make our way down yet another box to the third box down from the top, we see simple columnar epithelium. Simple columnar epithelium is found, perhaps most notably, lining much of our digestive system, particularly the small intestine, where it's specialized for absorption. If we jump down one box there to stratified squamous epithelium, we notice that our skin is an excellent example of stratified squamous epithelium. We have 5 layers to our epidermis, as you likely well know from anatomy, starting at the stratum masali, and our skin is a great example of stratified squamous epithelium. What we notice is the following, that if we look at epithelial cells, we can always characterize, at least in simple epithelium, these different sides that are possessed by epithelial cells. And so when we look at the diagram provided by our text, we see that this is a kidney tubule, and this is the lumen. If you can follow the pointer on the screen, this is the lumen of the tubule of the kidney, and then we see these cells on either side, epithelial cells, that surround that lumen. So on the far right, or at least towards the right, to the left of the blood vessel, we see the basement membrane, shown in blue there. Epithelial cells are anchored to a basement membrane, and so they're what we notice is the following. They extend up here and here. We refer to these as the basolateral sides, and so each cell has a side that is not attached to the basement membrane. It's not open to the lumen, and so we refer to those as the basolateral uh, membranes or basolateral sides of the cells. We also notice this, that each epithelial cell has an apical membrane. And so I'm pointing here to the apical membranes. If we follow this label in our notes here, we can see the apical membrane. It is the apical membrane principally that is specialized for either absorption or secretion and possesses various proteins that are involved in active or passive transport, moving substances back and forth into or out of the cell. We also notice this, that we may or may not have what are referred to as tight junctions that are located between the epithelial cells. The presence of tight junctions prevents the movement of molecules between the cells and forces any molecules or particles to make their way through the cell. This is often desirable for the cell because then it provides an opportunity for the cell to regulate what goes into the cell and what comes out of the cell. In other cases, there are not tight junctions, and so fluid, as we'll see, is permitted to move back and forth through these cells freely. We now spend just a moment talking about the last type of cell and the last type of tissue that we'll talk about, connective tissue. Connective tissue is there to connect, anchor, and support the various parts of the body. We've seen it a couple times today so far during the course of our discussion, including the fibrous skeleton of the heart, which anchors the atria to the ventricles. And what we notice is this, that students sometimes find connective tissue a little frustrating because it's not as easy to define as the three previous types of tissue. So for example, bone, blood, and dense connective tissue are all different types of connective tissue. Dense connective tissue is the primary component of tendons and ligaments. But what you notice is that these three types of connective tissue vary greatly in terms of their overall characteristics. To understand connective tissue, we need to talk briefly about the extracellular matrix. The extracellular matrix largely determines the nature of the connective tissue. The extracellular matrix consists of proteins and non-protein components, such as minerals or even water, very frequently water actually. And the nature of the extracellular matrix determines whether the connective tissue will be a liquid in the case of blood or quite solid, for example, in the case of bone. The extracellular matrix functions as a scaffolding for cell attachment. It enables the transmission of chemical messengers. For example, the hormones that travel, hormones are chemical messengers that travel through the plasma from one part of the body to another. And when we look at the components of the extracellular matrix, we notice that the proteins that we find there are often fibrous proteins. And so we see proteins like collagen. Collagen is one of the most common proteins in the body. And it's a big, tough protein. We find collagen in skin, as you're likely familiar. You likely find this familiar as a result of advertising campaigns trying to get you to buy things to prevent wrinkles that do things, or collagen supplements or something of that nature. We also find proteins that provide some flexibility for the connective tissue, such as elastin, the protein that provides some stretchability or flexibility in the nature of the connective tissue. We find non-fibrous proteins, perhaps most notably in the plasma. We find all sorts of proteins that are associated with things like blood clotting and immune function. We'll talk about those later in our course. But essentially what we notice is the following, that connective tissue, the nature of connective tissue is determined by the extracellular matrix. There are different proteins that are found in different types of connective tissue, although we do see that collagen is a very common component of connective tissue across the board. And then we notice that the non-protein components play a big role in determining the nature, the physical nature, how flexible that connective tissue is. If we look at bone, for example, hydroxyapatite is a mineral composition that plays a big component in terms of making up bone. In fact, we'll talk about the function of bone later in our class, and we'll see the role that osteocytes, osteoblasts, and osteoclasts all play in constructing bone. And what we'll notice is that these cells are laying down hydroxyapatite. Hydroxyapatite largely creates the solid, strong nature of bone. One thing that's always fun to do in anatomy classes is to take a bone and place it in a mineral vinegar solution for a few weeks. And what you notice is that when you pull it out, it's quite flexible because the hydroxyapatite, that part of the extracellular matrix, is now gone, but the proteins remain. So the collagen or elastin is still there, and it provides this bone that's very close to what you would think of as a toy rubber bone that can be bent back and forth quite dramatically. In section 1.3, we spent some time talking about the distribution of fluid in the human body. What we notice is the following, that water accounts for about 55 to 60% of the body weight of an adult male human, and that fluid is found in two compartments. The majority of it is found in the intracellular fluid, and so this, of course, is fluid that's found within cells, comprising the cytosol and the cytoplasm of the cells. Some of that fluid is associated with the organelles themselves, in other words. But a lesser volume, a lesser percentage of that is extracellular fluid. And so this is fluid that's found inside the body but outside the cells. 20 to 25% of that fluid is plasma, which may be surprising. This is, of course, comprising your blood. The majority of it, 75 to 80% of the extracellular fluid, makes up the interstitial fluid of the body. So if we look at the diagram found in the lower left-hand side of our slide, what we notice is the following, that the volumes that are found here are the volumes of the different fluids for what your book calls an average size human being, which they say is a 154-pound individual. Whatever the case, this is for an individual that weighs 154 pounds. And so this individual would have 3 liters of plasma, 28 liters of intracellular fluid, and 11 liters of interstitial fluid. Now, I think perhaps most important in this diagram is that we make sure that we're familiar with the idea, and we likely are, that interstitial fluid comes from the plasma. And so there's a certain amount of pressure that's distributed throughout our circulatory system. We get to learn about the specifics of those pressures in the cardiovascular system chapter. But that pressure leads to the exodus of fluid from inside the blood vessels to the surrounding area where I'm pointing here with the pointer, and that creates the interstitial fluid. And so you may ask the question, what's the difference between the plasma and the interstitial fluid? It's a good question, but it's a simple answer. The answer is not much. So the primary difference is the protein composition. As the fluid exits the plasma, in other words, as the plasma, you might say, leaves, the liquid portion of the plasma leaves, it leaves through openings that are so small that a filtering process occurs. And so the proteins that are part of the plasma are retained within the blood vessels, but the liquid is able to leave, and that liquid is the liquid that bathes the cells that surround the blood vessels of the body. Now, a good question at this point would be, well, where does that liquid go? Because it's continually leaving, and so it must go somewhere. And so I'm glad you asked. What we see is that this is where the lymphatic system comes into play. And so the lymphatic system is a portion of our body that's responsible for receiving that interstitial fluid and taking it back to actually the circulatory system. It becomes part of the blood once again. All over our body, we have, in addition to blood vessels, lymphatic vessels. Although, interestingly, this is not a circulatory system, but rather the lymphatic system in and of itself is a mechanism by which the interstitial fluid is picked up and then taken back, as I said, to the circulatory system. The lymphatic vessels merge with one another, becoming larger and larger and larger until eventually the two large lymphatic ducts empty into the subclavian veins, returning that fluid to the blood. It's also worth noting this, that the interstitial fluid, once it enters the lymphatic vessels, is referred to as lymph. There is no distinct difference between interstitial fluid and lymph other than location. And so it's the same fluid. There's no protein difference in this particular case, but once it enters the lymphatic system, we refer to it as lymph. Lastly, what we see is this. This is an example where pathophysiology, unfortunately, is helpful in illustrating the importance of normal physiology. And so the individual that you see in this particular diagram is suffering from a disease referred to as elephantiasis. This is a disease in which it's spread by a mosquito, in which a protozoan parasite enters the lymphatic system and reproduces in the lymphatic vessels, clogging the lymph nodes. And so blocking, therefore, the flow of that lymph and preventing it from making its way back through the lymphatic system and being emptied into the blood once again. And so I picked a picture that wasn't too disturbing for us, although we certainly feel compassion for that individual. But what we see is that there's a very distinct issue with the blockage of that lymph, and which just before it became lymph was interstitial fluid. And this illustrates the extreme importance of this circulation of fluid in our body. And here, when I say circulation of fluid, I'm not talking about the movement of blood through the circulatory system, but rather talking about the movement of plasma to interstitial fluid to lymph, and lymph back to plasma once again. On this slide, we want to emphasize the idea that all three of these different fluid components, we don't talk about lymph in this particular slide, but the three in terms of plasma, intracellular fluid, and interstitial fluid, all interact with one another to transport a number of items throughout the body. So, for example, what we see is the following, that in the case of the respiratory system, the point of breathing is to move oxygen into your blood. That oxygen is moved via red blood cells primarily out into the distant parts of the body. That oxygen is released. It moves very briefly through the interstitial fluid. It reaches cells, moving into the intracellular fluid, and then it reaches the mitochondria that are found within those particular cells. And so a similar but opposite process occurs for CO2. And then when we talk about nutrients and waste, it's also a similar idea. So you eat so that those food particles can be broken down, the nutrients can be transported into your plasma. That plasma transports it to the various cells of the body. Those nutrients move through the interstitial fluid, reaching the intracellular fluid of cells. And the opposite then, of course, happens for waste. And so what you notice in all of these processes is that the process of diffusion is distinctly important, very important in these processes. There is, for example, in the case of oxygen, there's no active transport of oxygen from the plasma into the cells, but rather there's a constant gradient that's maintained because the cells are constantly using oxygen. And so oxygen always has this tendency to diffuse from the plasma to the interstitial fluid and then into the cells. And CO2 being produced in the cells diffuses in the opposite direction back into the plasma. We do also, on this particular slide, make the point that extracellular fluid differs distinctly from intracellular fluid. We talked about so far the differences between plasma, plasma, interstitial fluid, and lymph. And we have noted that, as it says on the screen here, that those fluids, plasma, interstitial fluid, and lymph, are all very similar, with the exception that the protein concentration is higher within plasma. Now, however, if we look at the difference between extracellular fluid, collectively, those three, and intracellular fluid, we see that there is quite a big difference. Intracellular fluid contains different proteins that are responsible for regulating growth and metabolism. We see different signaling factors that are found in the intracellular fluid versus the extracellular fluid. And if you want to look at an extreme example, look at neurons. And so the entire functioning of our nervous system, our very, very beings, in fact, our abilities to think relies on the premise that our neurons, the 86 billion neurons in our brain, have the ability to maintain very distinctly different concentrations of ions, in this case, inside them, in other words, in the intracellular fluid versus the extracellular fluid. And so the transmission of any signal via the nervous system in our body is the result of movement of ions across that plasma membrane of nervous cells, and therefore a change in the intracellular versus extracellular concentration. So the take-home message then is that the composition of the extracellular fluids is quite common, but that differs distinctly from the fluid that we find within cells in the body. In other words, the intracellular fluid. A major theme in the discipline of physiology is homeostasis. Homeostasis is the process by which our body maintains various parameters within our body in a very constant state. So most of the parameters within our body are held within a reasonably constant state. So for example, blood pressure, body temperature, blood glucose levels, all three of those are typically maintained in a very constant state. And to be honest, although this becomes a dark discussion quite quickly, the body can't survive much fluctuation in any of those variables, and death results quite quickly if any of those fluctuate too dramatically in one direction or the other. So most of the common physiological variables of the body are maintained within this predictable range, assuming that the body is functioning in a normal manner. We do notice the following, that we describe these parameters, or at least the manner in which they're maintained within the body, as a dynamic constancy. So most of these parameters are going to vary a bit within a normal range, but the fluctuation varies greatly depending on the variable that you're talking about. For example, oxygen levels are extremely constant within the body. And most people, assuming that your respiratory and circulatory system are functioning in a normal manner, you have a great deal of redundancy. In other words, you have a great deal more oxygen found in your blood than is actually needed. But the oxygen levels are quite constant in a healthy person. So next, what we see is this, that blood glucose is a bit more varied. This is something that you likely can reflect on just from your daily experience in life. And so in the diagram from our text in the middle of this particular slide, we see these peaks in blood glucose levels that occur after breakfast in the morning, lunch in the middle of the day, and then a much larger peak, probably following a larger meal at dinner or supper, I suppose, at the end of the day. And so the release of insulin does a pretty good job of leveling out those blood glucose levels, but it's not instant. It's not as good a system as our regulation of oxygen within the body, and perhaps you can see the necessity of that. Our body has essentially zero tolerance for low oxygen in the body for more than just a matter of seconds. So if all organ systems in the body are operating in a healthy manner, then they're maintaining homeostasis, and good health is maintained within that individual. If homeostasis is not maintained, then pathophysiology results. Some sort of disorder or disease results as a result of the failure to maintain those parameters. Now, we want to talk briefly about how those parameters are maintained within the body. So we typically use the term steady state to refer to the state at which a variable is maintained within the body, and we understand that energy must be applied in order to maintain that particular variable. So for example, body temperature. A great deal of energy is expended to maintain a constant body temperature in the case of humans or any homeothermic animal, any animal that maintains its body temperature. However, we also use the term set point in this particular discussion. And so a set point is similar to the discussion we had a moment ago. The set point is you might think of it as the overall goal for maintaining that particular parameter, but we do see that the parameter that we're maintaining goes above the set point and below the set point before it triggers a return to the set point. And when it triggers that return, that's what we see occurring on the screen there. We see that it overshoots the set point and then goes back down and back up and back down. And this is basically what we expect to see in these homeostatic systems within a body. We introduced for the first time in our physiology class the term negative feedback. And so negative feedback is one of the core components of physiology. And it's the idea that if we have one of these parameters, we have a set point for the parameter, and if it wanders away from that particular set point, then certain activities occur within the body that bring that parameter back to the set point. So this is the most common mechanism of controlling these homeostatic parameters within a human body, or actually any the body of any organism. So let's look at this example provided by our text. And so these are our diagrams. Perhaps this is the first of these diagrams in our class, but we see these frequently, and these are very helpful to us in terms of our accomplishing our learning objectives in our physiology class. So we see at the top that we start, the room temperature goes down, and what happens as a result of that is heat is lost from the body temperature goes down as a result of that, and in response to that drop in body temperature, three things occur. Starting at the left-hand side, we see that we have constriction of blood vessels that are found within the skin. Your body, interestingly, has a tremendous ability to pull blood to the core of the body during times when it's cold, and therefore preserve heat. Just like conversely, when you're hot, you may have noticed, especially if you're fair skinned, that your skin gets quite red when you are hot. It gets red as a result of the fact that blood is being moved to those blood vessels that are close to the surface, and heat is therefore leaving your body. If we make our way from left to right, we see this. The middle response is a behavioral response. This person curls up to try to prevent, as we can see here following this arrow, heat loss from the body. And then if we go to the far right, we see that the person is shivering. And so this shivering is involuntary, and it creates heat production as a result of muscle contraction and helps raise the body temperature. And so hopefully the end result of all these activities is that the set point returns, or the parameter, returns to the set point. And so the body temperature returns to the original value. This is the idea of negative feedback. And we observe negative feedback frequently within physiology in the maintenance of various physiological parameters found within the body. We also notice that negative feedback may occur at a molecular level, as we see illustrated on this particular slide. In fact, negative feedback can occur at the organ level, the cellular level, organism level, or the molecular level. And so this is a common situation that you see in metabolic pathways within the body that you're certainly familiar with at this point in your biology career. Here we have a substrate that's transformed by enzyme 1 into an intermediate number 1. Enzyme B transforms this intermediate 1 into intermediate 2. Enzyme C transforms intermediate 2 into the active product. But you only need so much of the active product. And so what we see is this, that the active product goes back up to enzyme A and shuts down the production of enzyme A. It likely binds to an allosteric site. As you likely remember, an allosteric site is a site in the enzyme other than the active site, and the binding of this product to enzyme A changes the shape of the active site and prevents this substrate from binding to enzyme A. This works out perfectly because the cell only needs so much of this particular active product, and so as long as it has enough, then it's shutting down the action of enzyme A, preventing it from turning the substrate into active intermediate 1. But once levels of this active product drop, then they're no longer there to inhibit enzyme A, and this substrate can be turned into inactive intermediate number 1, and so the pathway fires up once again. So in other words, negative feedback can occur at the molecular level. We also notice that occasionally within physiology, something referred to as positive feedback occurs. So in the case of positive feedback, we have a situation in which the effector amplifies the changes that are observed. We see very few examples of this in physiology, at least human physiology, for obvious examples. We're typically maintaining these parameters within a certain range, and so positive feedback is going to push a parameter out of that particular range. There are two prominent examples in human physiology that we'll mention ever so briefly. The first one is the process of childbirth. And so in the case of childbirth, we see that the baby's head pushes against the cervix. This contact of the baby's head with the cervix creates the release of oxytocin. The release of oxytocin causes increased contraction of the uterus, and so that increased contraction of the uterus causes even more contact of the baby's head if it's oriented correctly with the cervix. And so this increases and increases and increases. You can see the positive feedback loop, but the end is, there is a distinct end, and the end is that the baby is born. Another common example that we talk about in human physiology is blood clotting. And so the details of this go beyond our discussion at the moment, although we do get to talk about them when we get to the blood chapter. But essentially, in the case of blood clotting, when you think about it, our blood is an amazing situation in which our blood is good enough to form a clot to prevent the loss of blood from the body, but it prevents that clot from spreading through the body and creating catastrophic damage as a result of the movement of that clot through the body. And so this is possible as a result of positive feedback. Positive feedback occurs in blood clotting when these cascades that involve multiple enzymes transform clotting factors into an active state, allowing them very rapidly to prevent the flow of blood from a blood vessel, assuming it's of reasonable size. Of course, this is not possible in all blood vessels. And then there's a distinct end to that that occurs once the clotting has completed. There are actually several factors that bring about that end, and we'll talk about those factors in detail when we get to chapter 4. It's not chapter 4, to the blood chapter. And so what we see is this, that most of the time in human physiology, that negative feedback is the mechanism that is regulating these parameters in the body. But occasionally, we see that positive feedback plays an active role in human physiology. Finally, we see that sometimes in human physiology, the set points that we talked about previously can be reset for one reason or another. That is adaptive. In other words, when I say adaptive, of course, I mean that it's beneficial for the organism. And so one example of this that we observe in human physiology is fever. In many cases, fever is adaptive because it allows the human immune system to work a little more efficiently at a slightly higher temperature. And although this certainly wouldn't be true for all bacterial pathogens, but it makes it a little more difficult for the bacterial pathogens to divide and continue the infection within the body. Not suggesting that fever is enough to shut down the division of bacteria, but it shifts the tables a little bit, in some cases at least, and provides an advantage for the human versus the bacteria that's causing the infection. And so fever is a resetting of that set point of body temperature, and so you have a higher body temperature, as we know, for a short period of time. It can be caused by different reasons. Some of them are internal within the immune system of humans. Others are external. And so as certain bacteria are broken down within the body, the components of the cell walls of those bacteria actually act as signaling factors that cause a resetting of the overall body temperature, triggering fever. We also notice that the body has some ability to participate in what's referred to as feed-forward regulation. And so feed-forward regulation is when the body responds when it anticipates a stimulus, but it hasn't received that stimulus quite yet. We see examples of when shivering, for example, may occur before cold temperatures are actually encountered, but when they are imminently anticipated. And we see it in the case of food. When food is anticipated, we see the body begin to respond and the digestive system start to kick in, even though the food has not come in contact, the molecules of the food have not come in contact with the body quite yet.